call. Thank you so much for joining the call today. I'd like to welcome Dr. Ben Moore. Dr. Moore is an outreach officer with Ensembl, and he's been generous enough to offer to do 2 sessions with us. So, of course, today's session and also another on October 12th to review all of the resources and tools available on Ensembl's website. So welcome, Dr. Moore. We really appreciate you being here today. And of course, as some of you may have already heard, Dr. Moore will be at ASHG as well if there are any questions. And there's also going to be an Ensembl workshop as well there. So thanks so much. And just whenever you're ready to get started, that'd be great.
Okay. Yeah, thank you very much for inviting me, Jenny. I've spoken with Jenny a little bit about the things that you want to know about Ensembl. So given our 2 webinars, we have 2 hours together, so we're going to have to try and cover as much as we can in the short amount of time. So I think I'm just going to begin without having much of a preamble. So we're going to be looking at the Ensembl Genome browser, the web interface, over the course of the next 2 webinars. So today I'm going to introduce you to Ensembl. Hopefully some of you are already partially familiar with Ensembl, but some of you might not be, so I want to talk to you about how the Ensembl data is organized. And then we're just going to jump straight in and begin exploring Ensembl. So I think one of the things that you particularly wanted to know about was the chromosomal location view. So we're going to look at what we call the region in detail. And we're also going to look at the genes and transcripts. So we're going to think about how Ensembl takes the genome assembly and annotates the gene models onto that genome assembly. The structure for the webinar 2 is a lot more simple. In fact, we're just going to be thinking about variation and phenotype data, as that's some of the themes that Jenny and I spoke about before as well. Obviously, if you have any questions that come up even outside of these topics, then I'll be more than happy to try and field those as we go through the webinars, which is my question slide. So I did pass on a link for all of our course materials to Jenny. Hopefully she has been able to disseminate that out to you beforehand, but if not, you can find them all through a website called training.ensembl.org. And in that website, there are links for all of our course materials, which include PDFs of our presentations and our course book as well, which contains screenshots of the demo that we're going to run through. So you can use that today, and it's also going to be available indefinitely in the future, so you can always go back to those materials as well. So just thinking about the objectives of our webinars, hopefully by the end you're going to know what Ensembl is, and then the different types of data that you can get with Ensembl. So we're going to begin exploring some of those in a little bit of depth, and we're going to be looking at all of that through the Ensembl browser website, so the web interface that we have. And one of the things that I'm going to point out along our journey is where to go for help and documentation. I think that's an important source of information that you'll probably be revisiting at some point in the future if you're using Ensembl. Okay, so Ensembl is one of a number of different genome browsers that are available. It's freely available and it's based on the web, so it's you need to have an internet connection, but other than that, you don't need much else to begin browsing Ensembl. And we need Ensembl and the other genome browsers to try and make sense of all of the DNA sequence data that is currently being produced at an astonishing rate. So without any context and annotation, it doesn't really mean an awful lot. So this is where Ensembl takes the genome assembly, we annotate it with different features like genes and transcripts, but also variants and regulatory features, and we try to make it accessible for scientists and users to get the most out of it to implement in their work. So we like to think of Ensembl as containing 4 main types of data. So firstly, we have our gene build. So this is where we take the genome assemblies and then we use biological data to predict the gene features that exist along the genome. The second type of data is the variation data. So this is where we annotate the genome with both small and large scale sequence variants, and we also have phenotype associations as well where they're available. The third type of data is comparative genomics. So this is where we compare both the genomes and the gene models between species to produce alignments and gene trees and make ortholog and paralog predictions, for example. And then our fourth type of data is the regulatory data. So this is where we take experimental data that have been produced by large consortia, such as the ENCODE project and the Roadmap Epigenomics project. And we predict the existence of regulatory features such as promoters and enhancers along the length of the genome as well. So to draw these four main branches of data together, Ensembl has a number of features, some of which we're going to get to cover in the next two webinars. So as I mentioned, we have gene builds now for actually over 100 different species in Ensembl. And then we take these gene builds side by side to compare them to produce our gene trees, which is one of the comparative genomics analyses that we perform. As I mentioned, we also have our regulatory build and our display of variation data. And when we're thinking about variation data as well, we also have a really useful tool called the Variant Effect Predictor, which we affectionately call the VEP. And this is a tool that allows you to analyze the functional consequences of your own variation data. I think we can allude to it, but we can think about the VEP if that's something that people want to have an example of using. Some other features of Ensembl is that you can upload your own data, which I think is a very useful feature if you're a scientist who's actively producing data. And then to access all of this data, we've got the web interface, but we also have some larger access methods. So Biomart is a tool that allows you to retrieve large amounts of Ensembl data for sets of features like genes and variants. And then you can also access data programmatically via either our Perl or our REST APIs as well. The best thing about all of this is that it's completely open source. So everything that I'm presenting to you, everything that you find in Ensembl is completely free for you to take, use, manipulate, change, and implement in your own work. And we really hope that you can do that as well. Okay, so I haven't changed this slide. It's meant to be over 100 now. We just had an Ensembl release where we included a lot more species. But these are the 80 species that we had just a few months ago in Ensembl. So you can see that we try and cover the broadest taxonomic space possible, while still including evolutionarily, medically, conservationally important species as well. So you can see we've got the primates, the four great apes and the primates. We've got rodents and other mammals, placental mammals. We also have marsupials and monotremes, as well as birds, reptiles, fish, and amphibians as well. You can also see that we've got C. elegans, the fruit fly, Drosophila melanogaster, and Saccharomyces cerevisiae, the yeast as well. So these are three important model organisms that we also have in Ensembl as well. So we're going to mainly think about the Ensembl project. And so everything that I've been talking about in terms of the number of species, these 100 species we have at the moment, these are all vertebrates. So Ensembl is a project that is aimed at annotating the genomes of vertebrate species, with obviously our other representative species as well. But we also have a sister project called Ensembl Genomes. So I'm not sure if this is going to be of any interest to you guys in your day-to-day work, but we also have genome annotation for bacteria, fungi, protists, non-vertebrate metazoa, and plants as well. And you can access all of that data through ensemblgenomes.org. It works exactly the same as Ensembl. So one of the important features of Ensembl is that we work on a release cycle, which means that every 2 to 3 months we package up all of the new data that we've been working on and release it all in one go. And this means that all of the data that you see is very traceable, since you can access the archives of Ensembl. So we all know that the biological data is changing, is very dynamic, and it changes what feels like day to day. So after an Ensembl release, for example, in December of 2016, we had the release of Ensembl 87. And then in the next few months, we took some new genome assemblies, did new analyses on our new data, updated any other databases that feed into our databases, and then we packaged that all up and released it in Ensembl 88. We're currently at Ensembl 90, and I'm going to show you how to access the older archives as well. If you're working with Ensembl, it's quite important, I think, to make a note of the version that you're using, as when you go back to it in a number of months' time, you can always access the data as you would have seen at that time as well, if it might have changed in the meantime. Okay, so that's my very quick introduction before I begin starting with the demonstration. Are there any questions at this point? No? Okay, so what I think I'm going to do is I'm just going to jump straight into the first demonstration that I have. And we're going to look at a region of the human genome, so it's on chromosome 4. And if you have your coursebook, you can find the screenshots for the walkthrough on page 6, but you can also follow along live with me or watch your screens as I do the demo anyway. Okay, so I'm just going to navigate into a new tab of my browser, and I'm going to go to ensembl.org. I'm just going to make my full screen. Okay, so this is the Ensembl homepage, www.ensembl.org. And maybe some of you are familiar with it already, but I'm just going to quickly go through some of the main features that we have on the Ensembl homepage. So at the very top, you can see we have this blue bar, and this blue bar navigates with you to whatever page of Ensembl you find yourself going to. So what you're going to see is that all of these links are available to you when you're using Ensembl. So the logo is actually a hyperlink back to the home page, and then we have links to our Blast Black tool, Biomart, which is this data export tool that I mentioned in the presentation just earlier, and then we also have links to all of our other tools that include things like the variant effect predictor as well. The downloads link allows you to access the FTP site, so you can find all of our data stored in flat files if you want to download genome-wide data. And then also probably one of the things that I really wanted to mention to you was the help and documentation. So I'm just going to click on this to start with, and this is the place where you should go if you have any questions about Ensembl. This is where we have all of our help and documentation, and you can navigate it using the menu on the left-hand side here. So I'm just going to use the hyperlink to go back to the home page, the logo. And then some of the other things to notice on the home page, firstly, is this light blue search bar here. So this search bar enables you to search all of Ensembl. So anything that you can think of searching, you can search here. So gene names, gene IDs, genomic coordinates, variant IDs, phenotypes, you can search them all in here and see what you can find for your species of interest in Ensembl. Over on the right-hand side here, you can see that we have information about the latest release. So you can see here that we're in Ensembl release 90, and you can see some of the data that we included in the release here as well. One thing that I wanted to point out to you further down the page is the View in Archive site link. So if you click it, you'll get a pop-up window that shows you all of our available web archives. So you can click on these different links, and they'll take you to the specific version of Ensembl that you're interested in. So you can obviously click, I won't do it now, but you can click on any of these links and it will take you back in time almost, so you can see the data as you would have seen it then. The last thing that I want to show you is just this Contact Us link at the bottom. So we do have a help desk in Ensembl, so if you do have any questions about using Ensembl, if you're confused about some of the data, for example, you can always email the help desk using this link here, and we'll get back to you helping you try to understand the data or answering any of the queries that you have. Okay, so going back to the home page again using the hyperlink, the last thing I want to show you on the home page are all of our species that we have here. So these are some thumbnails that have links to our most searched species, but you can also click View Full List of all Ensembl species here, and you can also access Ensembl Genomes through this link here. So this contains all of our non-vertebrate species if you click on this link. So I'm going to click on this View Full List of Ensembl Species. And what you're going to see here is all of the species that we have available in Ensembl. So what you can do is you can actually scroll down and have a look. But if you know the species that you're interested in, human, for example, you can use the filter at the top right-hand corner and find your species of interest. So what I'm going to do is just click on the name for the species of interest. And then what you're going to see is you're taken to a species-specific homepage. So you've got a search bar at the top here that specifically searches our human data. And then you can also see some other information and statistics about the genome assembly itself. You can see some of the other data that we have as well, the comparative genomics, the regulatory data, the gene annotation that we have, and the variation data as well. So what I'm going to do is just click on More Information and Statistics. And what you can see is we have some data about the different stats of the genome assembly and the gene build, so the number of different genes, for example, that we have. And you can actually see that we also have a link to older assembly versions. So currently Ensembl displays human genome assembly GRCH38, which is the current most up-to-date human genome assembly. But we also do have a dedicated website. If you're working with GRCH37 data, you can click here and it will take you to a dedicated site, almost analogous to an archive site, that contains the human genome data, but just with GRCH37 rather than 38. Okay. So again, I'm just going to navigate back to the home page using the hyperlink, the logo in the top left-hand corner. And what I want to do now is search for a particular chromosomal location using the search bar here. So the first thing that I'm going to do is choose my species of interest. So I'm going to select human from the drop-down list. And then I'm going to input my genomic coordinates. So we have a very specific format that we need to search for genomic coordinates if you want it to jump you to the relevant genomic location. Otherwise, Ensembl will just read it as a string of text, and it will just search for the numbers in all of our documentation. So the format that we need to use is firstly to put in the chromosome number. So we just put in 4. So we're interested in a region on chromosome 4. Then we need a colon. Then we need our start coordinate. So in our example, it's 122.868.000. And then we have a hyphen, and then we have our end coordinate. So in this case, it's 122.946.000. So we don't have any spaces, we don't have any commas separating the digits, and we don't have the CHR at the beginning either. So it's just a simple string of numbers here. So then you can click Go. And what this will do is it will jump you to what we call the region in detail page. So one of the things that you're going to notice is that we've got our blue bar. And what you're going to see as well is these tabs that begin to open up in the blue bar. So at the moment we're in the location tab. And as you begin to click on more things, you're going to open up more tabs. So when if you click on a gene, you're going to open up the gene tab. If you click on a variant to find more information about the variant, you're going to open the variants tab. And these are all open up sequentially in the blue bar here, and you can navigate back and forth just by clicking on the different tabs. So the next thing to notice is this menu on the left-hand side. So this contains data specifically relating to our view. So when you go to the different tabs, you're going to see that this menu on the left-hand side is differently populated because there's different data associated with that particular view. So now you can see that there are some comparative genomics analyses, for example, that you can access relating to our genomic region of interest. So these blue buttons on the left-hand side as well, these are also quite important, and I just want to point out one or two of them to you now. So the first is configure this page, and we're going to do this in a minute. So this is a page that allows you to manipulate the view that you currently see. So by default, Ensembl loads a small amount of data that we think is quite important and is generally interesting to everyone. But you're able to add more data and customize your view how you like it. So you can do all of this through configure this page, which is this option here. The next thing is custom tracks. So in this way, you can upload if you use this option, you can upload your own data to Ensembl. We're not going to have time to do that in our webinars, but you can explore adding your own data by clicking on this blue button here. And then the final thing I want to show you is export data. So if I click on export data now, what you're going to see is how you can export the FASTA sequence of this genomic region. So at the moment, you can see our genomic coordinates are already inputted here, but you can modify the upstream and downstream flanking sequences and the coordinates themselves as well. And then if you click next, you'll be able to download your FASTA file of the genomic region of interest. So I'm not going to download anything now, so I'm just going to click back, and I'm going to just exit the pop-up window by clicking on this tick in the top right-hand corner. Okay, so the next thing I want to show you now is how we can begin exploring the data on this page. So this page is divided into three main pictures. The first one is the chromosomal overview at the top here. So this is obviously human chromosome 4. You can see the label. And you can see this red box here. So this denotes our location on the genome. So this is the region of interest that we will begin focusing on the subsequent images. And then these other highlighted regions you can see are what we call assembly exceptions. So this is where there's been alternate assembly sequence that has been laid on top of the original assembly. So if there were any fixes or patches that have perhaps been added to the genome assembly since its release, then they come up here as what we call assembly exceptions. And you can click on them to find out how the original genome assembly is different in the assembly exception in this region, for example. So the next thing that I wanted to show you is the 1 megabase overview. So this is the second image on this page, and now you can begin to see some of the features associated with the genome assembly itself. So what you can see is that the data is now being organized into what we call tracks. So tracks is just a term that we use in Ensembl that just means a single type of data that can be displayed by itself in its own track. So for example, here we have the contigs track. And so what we see here is this light and dark blue alternating blocks. This represents the contigs that make up the genome assembly. So you can see this blue bar running through the middle is the genome assembly. And then we have the genes track underneath. So these colored boxes represent the individual genes, and you can see their names underneath as well. Again, we've got this red box, so this denotes our region of interest. And we can do a couple of things with this view as well. So we can slightly manipulate this using these options in the blue bar at the top. So we have our scroll to a region option, so you can click and pull the region back and forth to find regions that you're also interested in. Now what you have in the image underneath is an option to either update the image based on your new location, or you can reset the scrollable image, which means we jump back to our region that we were first interested in. If you change the option in this top right-hand corner to select a region, it changes your mouse functionality so that when you click and drag, you now drag out a new box, and then when you let go, you're given the option to either highlight the region or to jump to your new region of interest, which will then obviously change this subsequent image below. So I'm just going to get rid of this pop-up by clicking the cross. And then the final view underneath is what we call the region in detail view. So this whole view is our region of interest that we typed in at the very beginning. And you can see our data is now organized into tracks. So you can still see the blue contigs track. This is the genome assembly running through the middle. And then you can also see genes that are annotated on the forward and the reverse strand. So you can see this is the different transcripts of the FGF2 gene, and then the different transcripts of the NUD-T6 gene on the reverse strand. So what you can see further down, for example, as well, is that we also have some other data shown by default. So we have some variation data and our regulatory build as well. So if you're not sure about what any of this means, in terms of the color coordination, for example, you can see the legend at the bottom that will explain why the different features are colored in different ways. So the next thing that I want to quickly show you how to do is just configure this page. So if you click here, in this blue box on the left-hand side, you'll get a pop-up window that shows you all the data that you have at your disposal. So the first page that we see is this active tracks page. So you can see that this is all the data that we're seeing in this most zoomed in image at the bottom of our page. And then in different subheadings on the left-hand side, we have different options for the different tracks that we have. So we have different categories, variation data, for example, more variation data, somatic mutations, regulatory data, for example, as well. So what you can do, if you want to add some variation data, for example, you can click on variation, and you can see that by default we show all of the 1,000 genomes data. So you can see that this is a track that is currently in our view because the box is filled in. If you want to add a track, you just simply click on the empty box. So if we wanted to look at the 1,000 genomes from the American populations in the 1,000 Genomes project, we could just click on the box and then select our track style. So in this case, I would choose normal. If you're not sure about where your track is going to be, you can always search here to find your track as well. So you can search for 1,000 genomes, and then all of the 1,000 genomes tracks will come up for you there. And you can add them in the same way. If you're not sure what a track is, you can just use this eye. You can click on the eye, and it will give you a short description of what data you're going to see when you add that track. So when you've added all the tracks that you wanted, you can save and close the window by clicking the tick in the top right-hand corner. And then when the page reloads, you should be able to see the new data added in a new track. So what you can see here is this is the 1,000 genomes data we add by default. So this is all of the variants identified in the 1,000 Genomes project. And then beneath here, you can see this is the 1,000 genomes American variants. So these are the variants that were specifically identified in the American populations in the 1,000 Genomes project. Okay, so I think that is all I wanted to mention on the chromosomal overview, on the chromosomal location page, so the location view. I want to just quickly ask if there are any questions at this stage. Okay, so I can hear some ruffling, but I can't hear any questions, so I'm going to now move on to the second half of our first webinar. So I just want to quickly talk to you now about our gene annotation. So in Ensembl, as I mentioned, one of the types of data we have is our genes and transcript annotation. So in Ensembl, the gene models that you see are actually the culmination of two different gene annotation methods. So for every species in Ensembl, we have an automated annotation pipeline that takes biological data and makes gene model predictions based on this experimental and biological data. And then for a number of other species, 5 species in fact, human, mouse, pig, rat, and zebrafish, we also have manual gene annotation from the Havana group. So this is a group also based at the EBI that manually annotates genes for these 5 species. And so what you're going to see for these 5 species is the combination of the gene model that is the combination of both of these methods of gene annotation. So just thinking about the automated gene annotation pipeline now, as I said, it makes predictions of gene models based on experimental data. And what I mean by experimental data is nucleotide sequences, for example, cDNAs, that have been deposited in publicly accessible databases, so the INSDCs, the International Nucleotide Sequence Databases, ENA, GenBank, and DDBJ. The automated annotation pipeline also draws in data about protein sequences. So where we have protein sequences available from SwissProt and Tremble, which comes under the broad umbrella of UniProt, we draw in that data and use that in our automated annotation pipeline. And we also draw in data from the RefSeq project, which is based in the NCBI, and manually annotates protein and mRNA sequences as well. So this is the broad technique that our automated annotation pipeline uses for human, mouse, and zebrafish, because we have a large amount of data in these three different categories for these three species. For other species, we get a little bit more creative. So for example, one thing that we can do is to infer gene models based on homology. So we can predict genes in the chimp by mapping human cDNAs and proteins onto the chimp genome to make our gene model predictions as well. One thing as well that we also incorporate is RNA-seq data. So this is something that's becoming generated more and more frequently these days and is more and more available. And where it is available in these INSDCs, we use the RNA-seq data for the species that we annotate as well. So one piece of jargon that you might see when you're navigating and browsing Ensembl is GENCODE. So GENCODE is the name of the gene set that is made up of the Ensembl automated and the Havana manually annotated genes. So the gene set that is the merge of these two methods, the output is called the GENCODE gene set. And the GENCODE gene set is used in a number of different collaborative projects, such as ENCODE, that you may have heard of before, the 1000 Genomes project, and other projects as well. So this is a gene view that you might be familiar with looking at in Ensembl, and we would have looked at that very briefly in the demo just previously. So what you can see here is all of the transcripts, or some of the transcripts of the BRCA2 gene. So you can see a number of things based on the gene views, based on the gene model here. So the first thing that you can see is that each transcript has an exon and intron structure. So the filled in boxes are coding exons, and the intervening lines are introns. You can see the non-filled in boxes at the very end here and here, for example. These are non-coding exons. So these are going to be this is going to be the 3' UTR, for example, in these two transcripts. The next thing that you can notice is that the transcripts have different colors. So this indicates what we call the biotype. So the main biotype we have is protein coding. So this is the red transcripts, and then we also have non-coding transcripts, which are blue. So this includes things like nonsense-mediated decay transcripts as well. So we do have another color of transcript, which is gold, and this is what we call a merged transcript. So where the Havana manual annotation and the Ensembl automated annotation, where they both agree on the exact same model for a transcript, we color it golden. And this means that you can have a bit more confidence in that transcript's annotation, because obviously two independent methods have come up with the exact same transcript model. And obviously, you only see these golden transcripts in human, mouse, zebrafish, pig, and rats, because these are the only species that have the Havana manual annotation. One of the other tracks that you're able to add to Ensembl and view is the CCDS transcripts. So this is the consensus coding DNA sequence set of transcripts. This is where there's an agreement in the coding sequence of a transcript between Ensembl and Havana, the EBI and Wellcome Trust, and then also UCSC and NCBI. So these obviously only refer to the coding sequences. So you can see here we've got this golden transcript of ESPN, and the CCDS transcript is in green above. There's no UTR sequences at the end because we're only talking about the coding sequence. And then, so what we can see here is that this green transcript has been agreed upon as the coding sequence between these different genome browsing collaborations. So my final slide, or one of my final slides, is just to let you know that we understand that we annotate a number of different transcripts for a single gene. And so your question to me might very legitimately be, which transcript is the most biologically relevant? Which one should I use in my studies? Which one is the one to focus on? And we give a number of different flags and pieces of evidence that help you make this decision in your work. So the first is that we tag any transcripts as GENCODE basic, where they're part of the GENCODE gene set that have a complete transcript with a start and a stop codon UTR sequence. The next is we also give our transcripts a support level score. So a transcript support level score based on the quality and amount of data that has been used in the annotation of that transcript. So a TSL score of 1, transcript support level score of 1, is the best, which means that there is at least one non-suspect mRNA that has gone into annotating that particular transcript, whereas a transcript support level of 5 is of a lower quality. So that suggests that there might be some ambiguity about the data, there might be some missing pieces of data that underlie that particular transcript. The last thing is the denotion of a particular transcript as the APRI principal isoform. So there's an external group based in Spain, and they go through the transcripts for different genes, and they annotate the major isoform by combining protein structural information, whether a transcript contains functionally important and evolutionarily conserved residues, and they do some alignments between cross-species alignments as well. And they annotate 1 or 2 transcripts as being the principal isoform as well. So all of these different flags you can look at in the Ensembl genome browser as well for the transcripts that I'm going to show you in a minute. So this is my last slide. And what I wanted to quickly tell you is that all of our Ensembl genes and our features, all of our features, have what we call stable IDs. So where we have the names of transcripts that come from HGNC for human, our genes also have an 11-digit identifier. So it's always ENSG and then an 11-digit number for gene, and then T for transcript, P for peptide, E for exon, and R for regulatory region. And these won't change from release to release. So where we know that gene names can sometimes change, if a gene model stays the same between releases, the stable ID will also stay the same. So it's very important perhaps to make a note of the stable ID if you're working with a particular gene quite frequently. So for different species, we just have a suffix as well. So for mouse, for example, it's ENS, MUSG, and then the 11-digit number. Okay, so again, just want to quickly see if there are any questions that have arisen from my presentation in the last 10 or so minutes. No? Okay, so what I want to do now is move on to showing you the genes and transcripts that we have in Ensembl. So we're going to look at a particular gene called ESPN. So I'm going to search for this in Ensembl. So I've just jumped out of my presentation. I'm going to go back into my browser. The first thing to notice is that you can click on anything in Ensembl. This is something that I didn't mention to you before. This is all clickable. I guess you would call it as an adjective. So you can click on a feature if you're interested in it, and you'll find out more information. So you can see this transcript I clicked on, for example, is part of the NUD-T6 gene, which has this stable ID. This is the particular transcript that I clicked on, and that gives rise to this particular protein with a ENSP ID. And you can also click these links to navigate to the respective gene tab or transcript tab, which I'm going to show you in a moment anyway. So I'm just going to close that pop-up window and scroll back up the page and click on the link to go back to the home page. And I'm going to search for my gene of interest using the search bar. So I'm just going to type ESPN and then click Go. And then what you're going to see is the search results for ESPN. So by default, we always return human gene at the top, but you can filter the search results for different species or different features if you want to look for a more obscure species than human. But we're just going to click straight on the hyperlink at the top, ESPN human gene, to take us to what we call the gene tab. So what you're going to see here, is that we're now in the gene tab. So this is got the menu on the left-hand side that is got new data associated with it because we're now in the gene tab. You can also see that we've got the location tab next door. So if you wanted to view the chromosomal location that contains the ESPN gene, you can click here and go back to the view that we would have seen before in our previous demonstration. So some of the features that I think Jenny wanted me to point out to you on this page is firstly that you can see that there are links to some external databases such as HGNC. So you can see we've got the gene name from HGNC, and if you click on this here, it will take you to the HGNC pages where we have obviously drawn in that particular gene name. As you scroll further down, you can begin to see some other features of the gene. So the first thing that you can do is click Show Transcript Table. You may have already had this open when you first loaded the page, so you can just toggle the transcript tables being on or off by clicking the box. And what you're going to see here is a tabular representation of all of the transcripts of the ESPN gene. So these are the ESPN transcripts with their IDs, and then obviously you have information about their length, the length of the protein they encode if they're protein coding, and then the biotype. So you can see the red protein biotype that gives rise to particular proteins. And then you can also see links to external databases. So you can see this top transcript here, this golden one, which we can obviously already tell is a merged transcript that has been agreed upon by Ensembl automated and Havana manual annotation. But you can also see that there's a CCDS transcript associated with this transcript. There's a UniProt protein and RefSeq links as well that all map exactly to this transcript. The flags on the right-hand side, this is what I was telling you about with the transcript quality assessment. So you can see that we've given our transcripts a transcript support level score. So this one has a TSL level of 1. It's part of the GENCODE basic gene set, and it's been annotated as one of the principal isoforms of the ESPN gene as well. If we scroll down, you can just see that we have a graphical representation of the transcripts. So you can see these are all the transcripts you would have seen in the table, and this is the view that I had a screenshot of in the presentation. So you can see the exon-intron structure with the coding exons and the UTRs, for example. You can also see some of the other transcripts that have been annotated in the region as well. So what you can see is anything above the transcript, anything above the contig, this is on the fourth strand. Anything below the contig, this is on the reverse strand. Okay. So the next few things that I want to show you is just in the menu on the left-hand side. So we can, first of all, I'm going to hide the transcript table just to simplify our view. Oh. And then the first thing that I want to click on is sequence. So this is how you can find the genomic sequence that encodes the ESPN gene. And you can see you already have options to either download this sequence or to blast this sequence as well. So you can actually copy this sequence directly into our blast tool just by clicking the button here. And what you're going to see is that you've got the FASTA sequence with some annotation there as well. So because I've played on this page before, I already have some data that's being loaded onto my view, but you should just have the plain FASTA file, the plain FASTA view with the ESPN exons annotated in this view. So what you can see from the key is that the peach highlighting indicates that this is an exon in the genomic region, but the red lettering indicates that it's an ESPN exon. So what you can actually see here is this is the first exon of ESPN, because this exon that doesn't have the red lettering is an exon of a neighboring gene. Okay, so as we scroll down, you can see we have intron sequence, and then another exon, and you can keep going down. This is obviously an exon of another neighboring gene. So how you can add further data to this view is to click Configure this page. And you can choose to show variants on this view, which is something that I had already added. So you can click, it would have been no for you by default, but you can click yes. And you can also choose to change the flanking sequence and add some other annotations as well. And then once you've added what you want to, you can save and close, and this will change your sequence how you want it. Okay. So the next thing that I want to show you is our gene ontology annotations. I think this is something that that Jenny wanted me to show you. So you can see in the menu on the left-hand side, we have a link called Gene Ontologies, which is split into cellular component, biological process, and molecular function. So if you click on, for example, cellular component to start with, what you're going to be able to see in this view is all of the Go terms, the gene ontology terms, that have been associated with particular transcripts of this ESPN gene. So you can see, for example, these two transcripts of ESPN have been annotated with this Go term, which is the cytoplasm. So the protein product of these two transcripts has been annotated as occurring in the cytoplasm. And there are other transcripts that have been implicated in the cytoskeleton, cell projections, and the actin cytoskeleton as well, for example. So we have the same for biological process. So this is more to do with the actual pathway that and biological process that the gene is involved in. So when we can see here that the page loads, we can see that the ESPN exons have been implicated in the sensory perception of sound and locomotory behavior, for example. And then the same for molecular function. You can see that the ESPN transcripts are involved in actin binding and SH3 domain binding as well. Okay, so the next thing that I want to show you is also in the menu on the left-hand side, and it comes under external references. And I think this is going to be very useful for you guys as biocurators. So if you click on external references, you'll be able to see all of the different links to external databases that contain information about the ESPN gene. So you can see we've got the gene in Expression Atlas. We've got links to HGNC as well as disease databases, MIM, and NCBI as well, for example. And you can click on any of these to navigate to the other databases as well to get further information. So the next thing I want to show you, just the last thing on the gene tab, is the expression data. So if you click on gene expression in the menu on the left-hand side, this is data that's been taken directly from Expression Atlas. So Expression Atlas is a project that's based in the EBI and looks at tissue-specific expression of genes. So this isn't to do with the transcript, the different expressions of the transcripts, it's just the genes that we're thinking about now. So we've split them into male, female, and brain tissues, and we've got this diagram looking at the different tissues, but you can see the expression levels in the table on the right-hand side. So you can see the different shades of blue denote the expression levels that have been in the different tissues that have been looked at in these different expression projects that are all in the table on the left-hand side. So you can click and zoom in on this table to look at a particular region that you're interested in, and you can download this as well by clicking on the link in the top right-hand corner. Okay, so just in the last few minutes, I just want to quickly show you some extra information that you can find out about the specific transcripts. So if you click on Show Transcript Table and click on one of the stable IDs, so I'm going to click on the stable ID for the golden protein coding transcript at the top. this will take you to the transcript tab. So you can see here now we're in the transcript tab. So the menu on the left-hand side is going to contain information specifically relating to the transcripts of ESPN. So of ESPN 201, which is the name given to this transcript. So I'm going to click Hide the transcript table again. So what you can see here is the exon-intron structure of this transcript, and you can get the sequence of this transcript by clicking on exons in the left-hand menu. So here you can see the exon sequences and the intron sequences as well. So you can see the color coordination indicating either UTR or translated sequence in blue. And what you can probably see, because you might not have been on this page before, is that I already have the full intronic sequence and variant markup on my sequence. And how I got this was by clicking on Configure this page. So again, you can click on Configure the page, and you can choose to see exons. I had the variants in exons, and I also had Show Full Intronic Sequence selected as well. So this is how you can view, again, manipulate the sequence view to add data that you're interested in as well. So the next thing I wanted to show you is the cDNA sequence. So if you click on cDNA, you can see that we also have the cDNA and translated sequence of this particular transcript. So you can see in dark yellow is the UTR sequence, and then we have ATG as the first coding the first codon that gives rise to the methionine, and then we alternate with white and light yellow highlighting for the different triplet codons. So then we have an alanine and then a leucine, for example. Again, you can, with all of these sequences, you can either download or blast the sequences as you need to. The next thing I want to show you is the general identifiers. So if you click in general identifiers in the menu on the left-hand side, what you're going to see, again, is very similar to the external references and the gene tab. This contains all of the different links to external databases that have information about this annotated transcript. So you can click on these links and jump to the external databases, for example, in RefSeq, if you're interested in looking at the RefSeq transcripts. Okay, the very last thing I want to show you is information about the protein itself. So obviously we're looking at a protein coding transcript. So if I click on protein summary, what you're going to see here is a diagram that shows you the ENSP. So this is the stable ID for the protein that is encoded by our transcript. And the light purple and dark purple are the alternating exons. So this is exon 1, exon 2, exon 3. And then what we have here are a number of different algorithms that predict the existence of different domains and motifs. So you can see here we've got, for example, smart domains, superfamily domains, prosite profiles, and gene3D have all predicted this anchoring repeat in the N-terminus of the protein. And then there's also been the annotation of a WH2 domain in the C-terminus of the protein as well. So these are obviously predictive algorithms. Sometimes they're better and worse than each other at detecting different types of motif depending on the amino acid sequence that's being fed into the algorithms. Okay, so that brings me actually pretty much on time to the end of the webinar. I do understand it's been quite rushed, so I do apologize, but I would like to quickly afford you the opportunity to ask any questions if you do have any questions.
Yeah, I think you'd mentioned the variant effect predictor. I think I would love to see a demonstration of that and a bit more information on that. I don't know if that's possible next time. I think you maybe had it on the list. I think that would be really useful for people to see as well. Yes. But that would be great. Thank you. And this has been really, I mean, very clear. I think it has been a really great presentation. I did have one more question, but does anyone else have questions at this point?
So if there aren't any other questions, what I would really suggest is that I really only picked out some of the main pieces of data that, having spoken to you, I know that you guys are interested in. But there is also so much more data in the gene tab, the transcript tab, and the region and the number of different tracks that you can add to the chromosomal location view. So I would really advise you to just have a little play around, explore the different data that you can access in those pages. Obviously, if you do have a little play in the next couple of weeks, then I'm going to be available for questions. We can also we can do a quick follow-up at the beginning of next webinar to see if there are any questions that have come up in the meantime.
Yeah, that sounds terrific. And yeah, I mean, it really is amazing to see the depth of information on there. I feel like I certainly learned a lot today, so really appreciate it.
Okay.
Does anyone have any questions before?
Oh, this is Courtney. I just wanted to say I've used Ensembl a lot for the curations, and I definitely wanted to learn a little bit more. I know when you get to the cDNA tab, I use it often to try to make sure that a transcript in a paper is actually correct. And I notice with all your variants, I will use those, and I can go to explore this variant, and it will show you whether it has phenotyping data. It will show you all of the different chromosomal locations, ensemble mapping, as well as the NM coding for it. And I've definitely found that useful, but wanted to see what I could find from like the phenotype data that you're showing there.
Yeah. So I think what I'm going to next week in the next webinar, I'm going to talk a lot more in detail about variation data that we display. So you can what I've shown you in these sequence views is that you can add the variation data to the views if you're interested in it. And again, as you rightly pointed out, you can click on a lot of these, as I'm doing now. You can see that you can find out information about the different alleles, and you can jump straight to the variant tab, for example. We're going to do this in the next webinar, but you're exactly right. You can jump from this view, for example, to find out more information about more specific features as well. Yeah.
Oh, great, because I know I found that very helpful in trying to link back to Nomad and finding whether certain sites are, within Nomad and exact where it didn't coordinate. So thank you.
That's okay. So yeah, I think this webinar was very well. It was quite a lot to fit in, but next week or next time we do have a bit more. It's a bit more relaxed in terms of the amount of data that I need to show you. So yeah, I can be a bit more relaxed in terms of rather than rushing through everything, but we can explore the variation data and anything else as well.
That sounds great. Fantastic. Okay. Thank you so much, Ben.
That's okay. So Jenny, I just had one last question for you. Is everyone that attends today, are they also coming next time?
Hopefully. So all of our Biocurators receive the invitation. It really just depends on people's individual schedules. But I imagine that, yeah, many of us will be here on the next call as well. And as I mentioned, the fact that we've recorded the call today, for anyone who couldn't attend today, they can listen to the call if they're planning to, come next time. So would you recommend that if people weren't able to attend today but are planning next time, they should really listen to the introduction from today, I guess would be helpful?
Yeah, I think it would. I mean, you could also follow along if you have some use of using Ensembl before, some experience, but I think what we're going to think about in the next webinar does build on a lot of the things that we covered today already, so you might feel a little bit lost if you haven't watched this webinar.
Okay, so that's good to know. So I'll make sure that people are aware of that if they weren't able to attend today. Great.
Yeah. The very last thing to mention, I just have one slide that I want to show you. Just to point out some of the other help that we do have available to you. So we do have a YouTube channel with some recorded demos and webinars and things that you can watch. If you type Ensembl Help Desk into YouTube, you'll find it. There's EBI courses as well for some of the other EBI services like Expression Atlas and EVA and all of the different databases that you know about from the EBI. And if you do have any questions in the meantime about using Ensembl, you can please do email the help desk that I pointed out to you at the very beginning, and I'll be able to help you with any misunderstandings that you might have that have